Vaccines may contain different versions of the pathogen. Depending on what they contain, these vaccines may be more or less effective in inducing long-term immunity. Two types of vaccine include the entire pathogen, attenuated and inactivated. In both types, the pathogen has been modified to be less virulent. The attenuated live vaccine contains the living pathogen, but this pathogen has been weakened so that it's unable to cause disease. But the pathogen can still reproduce. This type of vaccine has several advantages. It's the most effective at inducing lifelong immunity, and when the pathogen is a virus, it will induce both humoral and cell-mediated defenses. The disadvantages are that the pathogen can cause disease in an immunocompromised person and occasionally can revert to its virulent form and cause disease even in a healthy person. An example of an attenuated vaccine is the MMR, which protects against measles, mumps, and rubella. The inactivated whole agent vaccine contains killed pathogen. Inactivation makes this type of vaccine safer. There's no danger of causing disease, but the killed pathogen can't reproduce and often has weaker antigens, so it only stimulates humoral immunity and often requires additional booster doses to gain full immunity. The vaccine currently used for influenza is an example of this vaccine. Toxoid vaccines contain no pathogens, only inactivated toxins from a bacterium. These toxins are treated with chemicals or heat to make them less dangerous, but this also makes the toxin a weaker antigen. Therefore, toxoid vaccines only induce humoral immunity and require periodic booster vaccines to maintain immunity. An example of a toxoid vaccine is tetanus vaccination. Genetic sequencing and recombinant DNA technology have allowed immunologists to delete virulence genes from the pathogen, thus preventing an attenuated pathogen from mutating to a virulent form. Subunit vaccines, such as the one used for hepatitis B, contain only non-pathogenic antigens of the pathogen, not the entire cell or virus. Thus, they cannot cause disease. Conjugated vaccines, such as the one for Haemophilus influenzae B, take the capsule polysaccharides of a bacterial pathogen, which are not very antigenic, and combine them with a highly antigenic protein to gain a more potent immune response. DNA vaccines are currently being developed that contain only the pathogen's nucleic acid. Thus, the pathogen's antigens could be made by the host cells, and they could induce immunity without ever introducing the pathogen itself.